So we have 4 panelists today that represent different points of view from industry. Well, they look like 3 right now, but the 4th one is on his way. I got an email from him a few minutes ago. He has an all-day departmental retreat in the electrical and computer engineering department, and he's just taking out 45 minutes to participate in this panel. So he will be here shortly. And my feeling is he didn't contribute any slides, so he doesn't get to talk anyway. So the panelists we have here, Matt Wiepert from Mayo Clinic. Matt, can you say a few words about who you are and what you do?
Sure, yeah. I'm Matt Wiepert from the Mayo Clinic. Oh, sorry, you want more than that, huh? So I'm a manager of a application development team. We do a lot of work supporting our bioinformatics area and our health sciences research teams, and mainly in support for the Center for Individualized Medicine. We support a very large medical genome facility which does a lot of next generation sequencing of many different types, and we're actually also moving those into the clinical practice. So we're collaborating with our Department of Laboratory Medicine and Pathology, which is where we have a reference range lab and a reference lab. And we also collaborate with external partners and data send-outs and just a whole bunch of stuff related to genomics, medicine, and individualized, personalized medicine.
Okay, great, thank you. The second panelist is Luda Mainzer, and I think many of you have seen and heard Luda already, but Luda, can you please introduce yourself?
Hi, I'm Luda.
That's it?
I'm a recovering bioinformatician.
I work in Victor's group, HPC Bio, at the University of Illinois, at NCSA. And we do research into high-performance computing in biological applications. So we think of hypothetical scenarios which potentially could be useful for the medical applications or non-medical bio applications. But these are research scenarios, not production scenarios.
Thank you, Luda. And finally, Paul Travis from Syngenta. Paul?
Sure. So I'm Paul Travis from Syngenta, and I think as you heard yesterday, Syngenta is an agricultural crop science company. I lead the HPC platforms and teams globally, which means I cover both the biotech space and the chem space in terms of provisioning HPC capabilities for our R&D community.
Okay, great. Thank you very much. So to prepare this panel, I put a few questions to the panelists, and they will try to provide answers to these questions, but my understanding is that Matt wanted to say a few words more about what Mayo Clinic does in this space, and
Well, I don't have to. I guess.
You don't have to. I'm just saying you provided me with a bunch of slides that I thought you might want to present. If not, we can skip directly to the questions, and if there's some time, maybe at the end you can give context. Paul also provided a number of slides that say a little bit more about what Syngenta is doing in this space. So the first question that I put to the panelists was, it's on the screen now, one of the oft-mentioned challenges faced by genome research is a threatening tsunami of data. And I'm sure that many of you have been in presentations where they showed, big waves and threatening skies and whatever. Perfect storm, I think, is what Paul was mentioning. And the question is, in your organization, has this actually hit? And what are you going to do, or what are you doing to deal with it? And what do you see as the data challenges for the next few years? And the first slide I have here is actually from Matt Wiepert, and maybe you want to elaborate on your answers to this question.
Sure, I can try and help with that. So the slides that I prepared were really just background on what we do at Mayo to give you some context for whether or not we're facing a data tsunami. So I would say that When we first started down the path of doing a lot of genomic sequencing, it was definitely focused on the research side, and it was focused on exome sequencing and really a lot of SNP panels, or looking for single changes using sort of array-based technologies. And we've moved into the whole genome sequencing arena, and there was a lot of initial freaking out, gnashing of teeth and so on about how much storage we would need, what was the hierarchical storage management aspects, how are we going to manage the data, how are we going to process the data. We didn't have a good set of algorithms on how to manage all this stuff. And if I were to fast forward from about 5 years ago to now, I would say I'm not really worried about the data tsunami, because we've got more infrastructure than we actually necessarily need to process the information for the research side today, and we've got enough storage and a really good sort of data management process to stream things through, age them out, and then archive them, and then have people get them back, and we've got some staff that are actually managing that stuff. So I would say that I'm actually, what I'm more afraid of are the revolutionary changes in the technologies that suddenly allow them to do sequencing about 10 times faster. And so that's always the fear that we have, is the infrastructure flexible enough, and are we building something that's generalizable enough that the algorithms don't change and then suddenly create a ton more data. And then the other piece of it that we're a little bit afraid of is our whole exome sequencing or whole genome sequencing happening on the clinical side. So what you see at Mayo is a lot of work and R&D going on to development of these pipelines that we then provide for our research community. And now we're moving them over to the clinic. So we've actually stood up 2 different sort of clusters, 2 different sets of hardware environments, and we've ported our software. And then once you move software into the clinical space, you have to do a lot of verification, validation, and make sure that that software never changes and that the outputs that you're getting are the same, so you can just continually run it. So it's good, because what they end up doing is putting a lot of scripts around our algorithms and controlling the input, whereas on the research side, we kind of always want to run it. Oh, can we tweak it again or change some parameters, rerun it, do some different things. You can't do that on the clinical side. You have to know exactly what you're getting and exactly how you ran it and exactly where the data went. So we've done a lot of really good work there. If they start to really do these things on the clinical side and we become a clinically validated testing there, I'd be very worried, because we have to keep the raw data 27 years or something, and this data becomes, incredibly scary, except for now I hear that we're going to get a petabyte for 25K, so can we take 5? And we'll just be good for, another 5 years or something. So again, every time we start getting really worried, it just seems that there's another thing that happens in the industry, or another partner, or another place where we can do this stuff. So again, I'm not too worried about the data tsunami. And we deal with it exactly like I said, with the PSP, or looking for outside vendors, or looking for other ways to work very creatively. People like Ben, actually, and Marie, and the team that provides our hardware support are the ones that really have to help. Because at Mayo, the way things are set up is I work in an application development area, which I can't develop applications if I don't have hardware, but if there aren't people developing applications, we don't need the hardware. So, you have that symbiotic relationship we have with them. And so they're very good at sort of listening to what our problems are and giving us something, but they also have to meet the needs of a very broad community and not just what we're doing. So this high-performance computing area is a very narrow slice with a very outsized proportionate need to what all of the other clinical applications might need. So again, outside areas can help us because we're outside the normal parameters for normal hardware and software applications that are running. So that's sort of what I see is when we go clinical and this becomes real, which it's going to become real, how are we going to find partners to help us run this stuff and not have it be run at Mayo, but follow clinical? We were a lot of discussions about business associate agreements, which means how do you share clinical data with other places, and how do those places that aren't used to dealing with clinical data suddenly say, oh yeah, we'll manage and treat this data like it was our own, except we're not used to clinical data and healthcare information, so it's a whole new world for a place like NCSA to think about, for sure. So those are some of the things.
Great. Thank you very much. It was a very thoughtful answer. Paul, I think, no, I have Luda as the next one. So, Luda, please.
There's no such thing as big data tsunami. We are the big data. And the media should stop dramatizing to get our attention by using the word big data tsunami. I think that the big data are here The tsunami is not scary. It's not going to hit us and erase us from existence. We should just embrace it, accept that we carry big data on our laptops, we carry big data on the cell phones, we receive big data in emails, hundreds of thousands of emails every day. That's big data. So, yes, big data are here and we should embrace it. So what form does it take to us in research bioinformatics? We look at it from the perspective of the size of input files, the size of output files, and the number of files. We have discussed this over the course of the meeting several times. For genomics, of course, the input files are a big challenge, especially when sequencing is being done on many individuals at the same time. Managing these data could be a challenge. Again, we've discussed this before. Managing is difficult due to the size of individual files and also because the sheer number of files can create bottlenecks for the metadata servers. Now, solutions already exist, actually, outside the bioinformatics space, outside the biomedical space. Again, we should just embrace them, adapt them to our purposes, and move on. The physics have been dealing with big data for a long time, astronomy, and so forth. And the scenarios that I posted on the slides are fairly standard. It's just we need to introduce little tweaks to them that are appropriate for biomedical space, including little quirks for the medical data and perhaps fewer quirks for non-medical biological data. You'll tell us about it later. But basically automating the hell out of dealing with the data, auto-streaming in, auto-streaming out, automatic archiving, indexing, recording the data. And of course, the straightforward approach is if you think that you're struggling with big data, make it be small data. There is research being done to accomplish this. For example, right now, the reason why the sequencing data are big files is because our genome consists of 4 letters that come out in various sequences. So we still represent them as letters. Every letter is 8 bits, but there's only 4 of them. Why can't we use binary format? We could shrink the size of the data right there. We could also compress the data better if we used binary format. We could learn to compute on compressed data as opposed to computing on the ASCII format that we're doing now. And the difficulty with these approaches is, even though they're being developed in research, because people have gotten used to using the old methods and the old algorithms, there is difficulty in adopting the new stuff. So actually, I suspect that the breakthrough will be in industry and in the clinic, where people truly have to manage big influxes of data, and the business constraints will require them to cut down the costs. And probably the adoption of these new computational technologies will be happening there first, is my guess.
Okay, thanks very much. Paul, this is your
Terrific,
first slide.
Yeah, I'll just go through a couple of these for this question. As a crop science company, we actually think that we have big data. We actually use the term, one of our colleagues from BioTeam, Chris Dagg, describes it as data that's bigger than you are. And I think we are finally starting to encounter that in the agricultural space. So what you see here is a slide of so underneath our HPC platforms, we have a transport service where we move data so that it's in proximity to compute. This is not all of our transport. This is the transport that comes through our help desk. Please reset my password, please move my data. So as you can see, we processed 450 tickets last year. We moved 180 terabytes, which, by the way, doesn't seem like a big number. I wish it was 0. But I think the key there is this is moving things like next-gen sequencing data to an informatics center so a scientist can work with it, a collaborator or a partner, what have you. It is simply a ticket that says move data from point A to point B. And then we have a data and compute fabric that allows us to move it. So I thought that'd be an interesting little way to start in that you can see we sort of are serious about data transport and that we literally have it within our help desk ticketing system. But what we're starting to encounter, if you go to the next slide, Victor, is we're seeing the world is changing. And we are changing in a way that I think sort of aligns us with what's been happening in the genomic space for a while. So if you go back to where the, let me go back to this, can I go back to this slide for just a second? I want to deal with the big data question. We do not think big data is next-gen sequencing. We handle that just fine. We do, our core foundation of our biotech business is sequence. And we can generate it, we can store it, we can analyze on it, we can do all kinds of things on it. We do not see that as our biggest data growth area. The next slide I'm going to show you is where we are seeing the data that's bigger than we are. So I'll say that. You look down here at the...
The one after that?
No, this one's good. This one's good. Okay. So we don't see next-gen sequencing as our biggest growth area. Where we see it is in the new initiatives that you are seeing in ag to go genome-wide, to look at gene-to-gene interaction, to look at gene-by-environment interaction, and to look at gene-to-gene interdependencies. These complex gene interactions are where we support our breeding programs to make sure that appropriate traits are expressed in our products, basically. So it's not just a one region of interest, one gene game anymore. We are doing pretty much everything at the genome-wide level in terms of supporting our product pipelines. So from a genomic standpoint, there's an exponential growth in the way that we are dealing with sequence. But more importantly, when you take it like gene times environment, gene times gene, we are doing a lot of stuff out in the field, if you will. We have aerial platforms that are doing imaging at very, very high resolution. We're doing digital phenotyping where we're doing measurement. We're looking at trialing data that's coming from a growing season. So you think of it, so is this, let's talk about that for a second. Trialing, how fast do we need to turn around trialing data? Harvest in the fall, plant in South America a few weeks later. We have to turn and burn on turning around our HPC jobs. But what happens is when you look at this, you add that all up, and you look field measurements, imaging, environmental data, weather data. You can be running at 6, 7, 10 terabytes per acre. You scale that to 1,000 acres or 100,000 acres, and all of a sudden, that is data that is bigger than us. So what that means is as that data is coming in, and why are we bringing it in? To look at the gene by gene and gene by environmental interaction stuff, is we really are requiring that to be pre-processed as it comes in. And what that means is that hub of an informatics center that we have always sort of lived by, it doesn't work quite as well anymore because we can't take that base level raw data set. Which is interesting because that brings up another whole philosophical challenge. We've never met a researcher that didn't want their raw data. They never want to throw anything away. In this case, you have to have policy where it gets thrown away. Partners create this analysis and results, and it feeds down the pipeline as well. So when you look at this, what you're seeing is you're seeing a change in the ag business and the way we do HPC. And what you're seeing is you are seeing big data streams coming in from partners. And they're being analyzed on the way. We may send data to those partners for correlation and additional analysis and modeling. And then finally, when it has enough intellectual property value, when it needs to have a certain multi-scale modeling component, we bring it inside. And then we basically bring it into Fort Apache and we protect it. We basically filter the data as it's coming down through to the point where we need to bring it internal, but we basically filter it on the way because we can no longer handle the big data sets that are coming from all of our providers. So to us, big data is real.
Oh, big data is absolutely real for everybody. That was my point. Big data are here. We just shouldn't be scared of it like it's a tsunami that's going to hit us. I mean, really, you know tsunami is coming. You know it's going to be big and deadly. Or are you going to stand around and be scared of it? No, you're going to protect, defend, grab a helicopter. I have a microphone right here. All right, okay.
So I think that, so the interesting point is you look at this graphic and you see it come into Syngenta Compute. And that Syngenta Compute is into that fabric that I talked about of compute and data. And within that internal, NCSA, we consider NCSA as internal. They look as like an extension of our grid environment. We look at them as a strategic partner. They are what gives us scale. We're hoping they will be the opportunity to let us really burst out and cover that trialing season, for example, so we can be ready for the next season's planting. That partnering with others to handle compute and data at scale is really what that Syngenta Compute block is. So this is different. If you've grown up around ag, you know we typically have dealt with chips and regions of interest and vectors and all this other stuff that we do to create products, and now we really have gone genome-wide, which is an exponential explosion.
Okay, thanks very much. We just had Professor Ravi Iyer walk in, and I know that he is multitasking today, so he is forgiven already. Ravi, can you say a few words about who you are and what you do?
I'm a professor of electrical and computer engineering and computer science. I also have an appointment in the coordinated science lab, which is just down the street from here, where it's really my home. I have broad interests in a whole range of analytical methods, particularly that address security, reliability. And the last couple of years, I think partly inspired by Victor, have become very interested in the analytics of genomics. And it's a consuming passion, if you like. I don't know how good we are at it, but we really enjoy doing it. Thanks.
Thanks, Ravi. I just want to go back to the first question that we asked and ask you if you have a comment on that. So the question was the following. One of the oft-mentioned challenges faced by genome research is a threatening tsunami of data. In your organization, has this tsunami already hit? What are you going to deal? What you're doing to deal with it? And what do you see as the data challenges for the next few years?
Great question, I think. I think we are hitting it. I don't know that the tsunami has actually hit us. You know, my good colleague here, Matt, may have a different sense because he's much more in the forefront. What I see as the challenge, which, you know, everybody, at least in electrical and computer engineering, computer science, says they do big data. But I really think genomics is the mother of all big data, and in the following sense. I think it's not just about sifting through this mass of data. It is really finding that nugget of truth, which is whether you have an oncogene or whether you have a particular disease. How does that combine with, how can you combine that with one's genetic history? Plus, how do you combine that with the environment? You know, do you live now I hear the New Delhi is highly polluted, one of the most polluted cities. But do you live in New Delhi or do you live in the mountains in Utah? What we see is that these other factors make a huge difference. And how do you bring it all together, I think, is a consuming challenge. And whoever has a handle on it, I believe, is going to be transformationally successful in industry, of course, in academia. But this is a big challenge for us in computing. And to this end, we are actually building a new machine that we think will accelerate this process as well as speed it up. We do extraordinary measurements, working closely with what my students say, Luda and company, because she's so inspiring to the students. So we have a great program, I think, and great set of colleagues to address it.
Thanks very much, Ravi. We can now move to the second question that I'm putting to the panel, which is, more kind of practically oriented. And the question is the following. Many hardware and software vendors are touting turnkey solutions for managing and analyzing genomic data, mainly the healthcare context, but also more generally. How do you feel about this, both in terms of the solution's functionalities and of the transparency of their underlying methods? And finally, have you had any direct experiences that you may want to comment on? And I'll let Luda lead the way on this one.
Yes, so the question was, what are my feelings? My feelings are frustration, annoyance, and company.
It's a normal state of affairs for you.
It's pretty normal state of affairs for me, that is true. However, it is particularly aggravated when I think about this question. So why is this? Yes, we have had some experiences with certain companies or individuals who think that they have this great new software and they want other people to buy into it, to use it, to get license for it. There are a number of different models that they propose. Sometimes you pay for license, sometimes you pay an inordinate amount of money for support, sometimes you have to actually get a box, which is a hardware piece. Either way, it usually has to cost a lot of money because they promise a speed up, in this case, in the genomic variant calling workflows, going from 24 to 30 hours down to under an hour. Okay, so this is a big deal. That's a big speed up. Okay, so if you think that you can promise me an analysis per genome under an hour when now I'm spending 30 hours with extreme parallelization on the latest and the greatest in HPC developments, which is Blue Waters, yeah, that is a big deal. I understand why you want me to pay big money. But if I need to pay big money for anything, wouldn't I want to think about it like really hard? Which is why it seems logical to me to first find out what the software really does and whether it does what I want. So I want to validate it. We're in research, so we're not going to pay any money for anything. So we make it our business to validate it so that other people could make decisions, being more informed. So, when things like this arise, the first question we ask is, tell us the algorithm. We understand the algorithm in the current open source software. We think we understand it pretty well. The folks in the IR lab spent a great deal of time studying the source code and figuring out how to improve the algorithm and so forth. So we think that at the UIUC, we have a great deal of expertise on the algorithm end. So tell us, what is your algorithm? I understand you want to keep it proprietary because you don't want to give away the opportunity to earn money, but please at least tell me in the broad strokes. And what seems to happen is, oh no, the algorithm is so difficult to understand. No, let's just not talk about it for now. So now they want to patronize me because they think I won't understand their algorithm? No, that's not going to work. If I don't understand how your shit works, I'm not going to use it. Okay. Then they come and say, well, you have to go and test it on our box. Or even better, you have to test it in our cloud. So now I need to use their data in their cloud to test their software in their cloud, and I have no control over what's going on there, which means they could have pre-computed stuff. They could be lying to me and wanting me to pay big bucks. No, that's not going to work. I want this software under NDA on my hardware with as much stuff open as possible so that I could validate it. If you want me not to talk about it, no problem. But I need to know what's in there, because otherwise I can't recommend it to other people, and I certainly will not be paying money. The other two actually answering questions and stringing us along, we have had experience with this. We have evaluated some software that was produced by a company, and it was actually placed as open source. We got some unflattering results. We thought, well, before we publish the unflattering results, maybe we should give a call to the company and ask them maybe we didn't do something right. So we sent a bunch of emails. They responded and said, well, you didn't use the newest, latest, and the greatest of the versions of our software. Okay, no problem. Send us the latest and the greatest. Let's sign an NDA. Oh, okay, we'll get back to you. And they were getting back to us, and they weren't getting back to us, and finally, we never got the latest and the greatest of the software, even though they said that technically they didn't need an NDA with us. So finally, don't string us along, and actually answer questions in a reasonable, respectable way, as opposed to saying, no, it's too complicated, you won't understand, it's not going to work.
Thanks very much. I can add to this that some of these companies have business models that can be rather surprising. One company asked us to pay them a license fee of $20,000 a year or something like that, plus pay for their people to come tell us how the software worked. And while what we were doing was actually a service to the company. So I told them our business model is to bill you by the hour for the time that we spend testing your software. And this is going to add up to exactly the same amount that you're charging us. Is this really what you want to do? And that actually solved the problem. Matt?
Sure. So I think I want to say that I was a little myopic in my first answer. If you were to tell me about what we do, generally I was thinking about the genomics throughput pipeline. If I start expanding into what is it that we're going to be doing with the data next, it becomes a whole another issue. And one of the things that this question gets at in terms of commodity genomics tools is that there aren't any. And part of the reason for that is just as she's saying, we're still evaluating our algorithms, and if we're evaluating algorithms, which means that we could have 3 different groups running 3 different types of algorithms or tweaking them in different ways, then we've not yet actually hit the point where, it's just like a normal instrument test that we run in a lab, that we never question what actually happened on the inside of the instrument. I get a blood gas test, I do it again, I get the same result every time. I could take the same data set, run it twice, and get slight variations in what differences we saw in the genome. And so right there, I already know that we've got problems because that's not going to work. And so the bioinformaticians, like Luda, tell me that they don't have any level of confidence that there's a real commodity thing. There's consensus and agreement that, we kind of run these sets of tools and we sort of agree on them. And so we kind of go down those paths, and that's really good. I think I won't mention vendors, but Ravi and I were talking with a few people, I think it was last week, and the question was, well, they want to package up and make end-to-end sort of genome tool pipelines. And then I kind of said, well, which pipeline are you going to use? And I said, well, we are actually running our own pipeline here at Mayo. It's published, Genome GPS. That's what we did in research, and that's what we now have over in our clinical side. And well, I don't know, if you guys were to sell something, I'd kind of have to get bioinformaticians to agree, and then they would test it and do validation and see if it was the same input and output. And I don't know if I'd be anywhere further, because then I'm locked into this vended application where I can't tweak and change anything. So that would be a little disconcerting, but not to me. We're IT, we can do anything. It's just money and time. We don't care. So, really what I worry about then is on the clinical side. I think I was hearing from my colleagues a little bit about one of the vended applications that we use today that charge by the core, that then we threw that on PSP. Okay, well, now we can't afford to do what we did any longer based on the licensing agreement. So the thought process of the level of computation versus the type of hardware that's being provided today doesn't match. And that's just discussions with vendors and reasonable prices. I mean, I understand it's a business, they got to make money. And we want to treat our patients, and there has to be a balance in between that somewhere. So I definitely don't see transparency from the vendors. I definitely don't necessarily see consensus on the right tools, and that's kind of my answer.
Thanks, Matt. Paul, you want to comment?
Sure. So our HPC sysads, they touch about 185 applications. So that's sort of our universe. Probably about a half of those are commercial packages. We are heavily loaded on the cheminformatics side, the computational chemistry side, in terms of the commercial packages, because it's an older, more established industry. And I will tell you, in that space, licensing kills us. When we try to go to scale, the cost for scale is a hard business case to make. Now, what's interesting in the biotech space, which is a little newer, if you will, we're seeing a different challenge where we still have commercial packages that we use. We still pay license fees. We still look for those perfect fits, if you will. But there are two other things going on that I think are interesting. One is a lot of people coming into the industry, particularly coming out of universities, come into our company with standard tool sets. And that might be the statistical package R, or it might be that they love MATLAB. There's a variety of those. And I think the value proposition associated with some of those is sometimes problematic, put it that way. We seem to pay a lot to let them have their favorite software, honestly. That's the first trend that I would say. The second trend that I would say, particularly in the biotech space, that we really like. So in the chem space, we run one workflow engine. It's a commercial engine. It costs us a ton of dough every year. In the biotech space, we use Galaxy. It comes out of Penn State and Emory. Has a wonderful bioinformatics toolshed. It links to our data sources. We're part of a 30,000 user community. It is a total win for us, and it's a different paradigm for software delivery. And I think we're seeing that there's case after case, particularly in the biotech space, where some of the commercial companies are getting a little bit of a run for it and producing some really good platforms.
Thanks very much, Paul. That was very insightful. Ravi?
So I'm going to maybe strike a slightly different note. The simple answer, I think, is the tools that are there don't agree with each other. In fact, one of our students working with Mayo, working with Luda and Victor and HPC Bio, has developed a benchmarking tool, which when you look at the output for the same set of mutations that across the board, the results are quite all over the place. And I mean, in a sense, that's a cause of worry, although when I go and talk to the biology colleagues here at Illinois, they have their favorite tool. And it might be that it is GATK. And doesn't matter how slow it is or whether it just runs on one machine. They say, well, even if I don't get the right answer, I get some insights and I can go and fix it. And maybe, and when you say, well, that's fine today, when you have maybe a few genomes to experiment with, what will happen when you have lots and lots of data and they are not quite ready for it? There's another factor in this, is that when you go and say, for example, determine the variance, what do you go and use it for? And again, it was very revealing in the work in our collaboration with Mayo, when the physician or surgeon at the other end of our phone call said, Ravi, you give us maybe 100 variants. It's of no use to me. I have a patient sitting there. I can't go and tell her or him, you have potentially 100 different possibilities. I have to give him maybe 2 or 3. What can you do to really come back and give me, with high confidence, a small number of variants that I can then go and really get into and draw upon my experience to go and give some advice to the patient? And the time frame is that a patient comes in on a Monday, and if I'm not wrong, by Thursday, the Mayo team has to tell them, they meet, of course, the family meets, or the patient and whoever, and they have to give them the results. And these people have to take it, and they go home or do whatever with it, and have to come back for treatment. So 4 days is the time they have, not just for us to run the code, but to run the code, make the inferences. That means algorithmically look at the patient history, patient monitored data, the genomic data, the genetic data, the external factors, provide, and with a human, very clever, very smart human in the loop, provide very focused and high confidence decisions. Where do I think it'll go? I think in the end, if the work that we are doing, that here is, and other places works out, it will become commodity. It has to be commodity. It'll probably be in the form of a specialized hardware, a chip that has a very similar role as GPUs, but I think you probably have lots of memory in a stack very close to it, and people like Wen Mei, who's here, build the software stack and the compiler to make it all work. But I think it's at once a serious computer engineering, computing problem. At the same time, this has to be done very closely working with bioinformaticians, biologists who really understand and have the insight.
Thanks very much, Ravi. I think that the consensus here is that currently there is, in this space, there aren't many solutions out there that we can really feel comfortable about using in production. And I'm amazed to see that most of the vendors really tout mostly their user interface rather than the actual performance of their software or hardware.
It's absolutely true.
Anyway, we have another a little over 10 minutes in this session, and I think it's going very well. And the last question I had was about time sensitivity. So the question is, how time sensitive are the analyses that are being performed in your organization, and what is the relative importance of accuracy versus time to solution? And so also, how far are you willing or planning to scale up your infrastructure to match processing time to your logistics requirements? And I know that logistics requirements are very different depending on which industry you're talking about. So maybe, Paul, you can start on this.
So let me start with the end of the question first. Building out infrastructure. We will invest in infrastructure to support proof of concept and pilot and discovery. We will invest to figure out what the new workflows are, work with the tools, that type of thing. Scale is important to us. We cannot go fast enough to support our R&D community. So to support those initiatives, we really look to have contract capabilities, contract grid, if you will, technical consultancy partnerships. I mean, this is NCSA. This is the classic, where they give us not only a grid capability, an infrastructure extension, if you will, but there is a deep bullpen of consultancy around it to help us optimize. That is sort of our future, I think, in terms of trying to meet those time demands. Our curve is a big, scary curve, and there's no place in the future where we see it flattening. So we're going to need to build good collaborations, partnerships, and that type of thing to be successful. But the base piece, we do have our own computational laboratories where we build the new workflows, where we put in our things. One of the things we're seeing is, and I think the sort of underlying thread of all my stuff is we are seeing real benefit to data and compute proximity. And if you look at what we actually are doing in our laboratories with the Hadoop platforms, MongoDB, faster backbones, file systems, it's all about how can we get compute and data to go faster.
Yeah, so in other words, the opposite of the cloud.
I didn't say that, but yeah, that can be sort of inferred.
I'm just putting words in your mouth. Maybe, Matt, you want to, I think you have a slide here. Yes.
Sure. So the answer to this question is it's time sensitive and it's not, and it depends on the case. So let me run down the case where it's incredibly time sensitive, and it would relate to the sorts of things Ravi was talking about. So at the Mayo Clinic, we started some individualized medicine clinics, and we have a couple of different service lines, as we call them. And one of them is related to cancer, as an example. And when people come into this service line, basically they're refractory to care, meaning that we've tried everything that is normal clinical practice, and the patient's basically not being cured and on a very scary path towards not being alive much longer. And so, they can come to us and they might have 3 months left. So I can't emphasize enough that, we better make every bit of this time sensitive. So they'll come into this clinic, and what we're trying to provide to them is genomic sequencing of their normal cells and their tumor cells and looking at differences, and then trying to map that to any other kind of treatment that's out there. So we understand that there's lots of drugs out there, but if we look at the genetics of what this patient has and some drugs that had worked with different types of diseases but had very similar genetic profiles in terms of what that drug and the pathway that drug was affecting, you might then try off-label drugs, and you might want to try them very quickly. And so, we want to get this data very, very quickly, and that means, if it's a month and it's a 3-month lifespan, if you don't do something to a person, that's a third of their lives. Yeah, I would like that data back tomorrow. Thanks very much. So you can see that becomes very time sensitive. And then we're trying to do lots of different interesting things to pull in external data sets to analyze with the genomic information, and you can start to imagine the analytic platform that you'd want available to you to pull in every bit of information from every external and internal database and annotate the crap out of what you have and make that sort of make a lot of sense. So yes, very time consuming, and we still don't have good visualization tools on top of that, and so that then makes it a lot harder. We have some tools, but we really need to be working on how you can make data and information pop to the consultants that are looking at this data to give some advice to the clinicians that can then share with the patients and make something happen. So research, okay, if the system's down for a day or two, there's a much different set of parameters. So there, it's time sensitive because the researchers want their data, but it's not nearly the same level of need that we would have, but still kind of important. I think another part of the question was accuracy. I don't know, that's yet to be determined because we use an algorithm called Novaline in our pipeline, and that was something that actually caused some problems when we were working a couple years ago back with Luda and her team, because that's not open source and it's a licensed software. But the reason we used it at Mayo, even though it ran longer, it was like 0.5% better. And so a lot of people internally, at Mayo, the bioinformaticians were fighting about it, but the guy that was in charge said, no, it's 0.5% better, even though it was not well understood and we couldn't tweak it. So, those are the weird decisions that get made. So it depends on who's making the decision as to whether it's accurate or not, or whether it's the right thing to do. And we're willing to scale up to the needs of the business and their tolerance to pay for what they need. That's why we're here, because we don't think we could provide the right level of hardware necessarily to the clinic in our own data spaces, plus given the variability of what we might need to do to change things in and out. If we spend the capital and own it ourselves, the proton beam was a perfect example when Ben went through that talk, because we had all kinds of flexibility to try all kinds of things out, and we had nothing at the Mayo Clinic, and we didn't have anything to deal with. That capitalization versus operationalization piece is really critical. So we're looking for all kinds of ways to do that, and I would think that we're willing to scale up, but we have to do it at the right price point for the bang for the buck for your patient. So,
thanks very much. Ravi, you want to comment?
Sure. I think it is time sensitive. Whether you are a clinic like Mayo or MD Anderson or what have you, providing this as a service, because people who come there usually have been to their local clinics, to their local call clinics like here in Champaign, and then it hasn't worked, and they go to Mayo as the next level, and they need results fast. But at the same time, we really need to get a handle on accuracy. And what we increasingly find is that both industry, that's companies that are coming out and saying, I can do this in 10 minutes, I can do it in 15 minutes. We can do now the haplotype caller in 23 seconds, but really, how accurate are you? Where are the repeats, and what are the areas, the unknown areas, and how are they going to influence your answers? I think they are very important questions. So on the other side, I think it is time for new algorithms that take advantage of the fact that in this kind of when you analyze genomic data, when you analyze history of the patient history, the patient's genetics, or go through and see how a particular seed, and we had a call from this company, seed company in India, saying, well, there's so many different climates and environments, and how can we customize our next generation seeds to work well in very arid climates as well as tropical climates. You have to really think about what are the better algorithms. What is the right computation model to go and produce accurate answers for these, in these cases, as well as do them very fast. And that, I think, is happening. It certainly is happening here. I think it's this joint effort to combine these two into, hopefully, into a single metric that we can benchmark, will be transformational to industry. And we see in our center that we have for computational biology and genomic medicine, the companies that are interested across the board in these two parameters. On one hand, of course, there are healthcare companies and healthcare providers, but the other spectrum is agro-businesses. And in the middle, you have pharmaceuticals, you have others, the healthcare providers, and a wide spectrum of people who are interested. And the answers are not always the same. The accuracy level is, and the speed with which they need the answers is not always the same. So somehow, I think we have to take the methods that we reproduce and allow the user to tune the benchmarks, to tune the system in such a way, or tune the software in such a way that they can get the level of accuracy and level of performance they want. I think that's where the sweet spots are different, depending on who the constituents are. And I think that's where the challenge is. It's not just a challenge for us. I think if I think of you as being industry, I think this is the future. I mean, it's going to be, and as we are speaking, I can see that it's transforming the computer industry for sure. You know, when IBM, Intel, Cray, Microsoft, all of these people come, and as soon as you raise the flag of genomics, you know that this is part of their future business. So I think this is where the excitement is. I think this is where the sweet spots in investigations are.
Thanks very much, Ravi. We are running out of time, unfortunately. I have Luda's last slide up here. And Luda said, maybe, Victor, you should comment on this. So I will do it very quickly. It was mentioned, I think, yesterday or the day before, that we are working with people actually at University of Cape Town and our two representatives from CHPC. CHPC is also the Center for High Performance Computing in South Africa. It's a different kind of time sensitivity. Here we have a very large bolus of data that's going to be coming in, and we have very strict timelines for actually analyzing these data. And the goal of the project is to design a genotyping switch chip that can be used for looking at genotype disease associations in African populations. And there are actually a number of research projects all across the African continent that are waiting for these chips to actually become available. So that's why there is a tight deadline on the design, so they can actually genotype all these people. And they have literally tens of thousands of patients enrolled in various clinical trials across the continent. And what this slide shows is just that we have to analyze about 350 high coverage genomes, which probably for Mayo sounds like, you know, a rather trivial number given their current patient load. But even though we have done a lot of work on looking on a semi-theoretical basis how to scale up and speed things up, now we're suddenly faced with a production problem. And it's like moving from research to the clinic for Mayo. All of a sudden, this becomes time sensitive. And, I'm not going to comment on all the steps and so forth. I just want to make a point that when suddenly you get a project like this, it lights a fire somewhere, maybe in the back. We, even in research, have to deal with sensitivity to timelines. Okay, that's all I'm going to say. I thank all of you for being here, and I certainly thank the panelists for really nice presentations.